Hello, everyone. Thank you for joining us today. We'll be presenting our monthly CGC webinar, and we are featuring today an honored guest, an office hour favorite, the newly minted Dr. Vaidhyanathan Mahaganapathy. So, Vaidhi is a recent PhD graduate from Rutgers University, and he now works as a computational biologist at the Ellison Institute for Transformative Medicine. In this webinar today, he's going to summarize his work with Dr. Khiabanian's group on developing the MERIT pipeline on the CGC. So MERIT, which stands for Mutation Error Rate Identification Toolkit, was originally developed by another lab member, and it's a multi-step pipeline that utilizes Samtools to identify positions, all positions with alternate alleles from aligned reads. It then extracts additional information from these variants. So thanks to Vaidhi's hard work, this tool has now been optimized to run on the CGC platform, and it will be made publicly available to the CGC community in the coming days. So we're working on making this publicly available so that everyone is able to do it. It's a really complex pipeline, but you'll be able to just run it with a simple click of a button. And Vaidhi will talk more about his work. So I'm just going to hand it over to him, and he can begin whenever he's ready.
Thank you, Rowan, for that wonderful introduction and making my life easier with a good background on MERIT, so that when I start yammering about what it does, it's going to be easy for people to follow. Thank you. And it's also nice to be back at CGC after my brief hiatus. I do pop in on, plan on popping in on the office hours once in a while. Good afternoon, everyone. So the topic of my dissertation is a computational framework for detecting low abundance clonal hematopoiesis mutations in large-scale tumor sequence datasets. The way I've structured this talk today is to first explain what CHIP, or clonal hematopoiesis of indeterminate potential, is. Why are we interested in studying CHIP? What was the method that we designed to study CHIP? And in that process, what all we broke and what all broke by itself, happens all the time. How did we go about fixing them, and were we successful? And finally, sort of talk in the context of why this is relevant, and in particular, relevant on the Cancer Genomics Cloud platform. So clonal hematopoiesis, or CH, is a population of hematopoietic stem cells that share an acquired gene mutation. This is a characteristic of myelodysplastic syndromes and leukemias, and in some older individuals who have no hematological malignancies, a single driver gene is usually mutated to confer the CH to a set of clones. When this phenomenon happens, it's usually referred to as clonal hematopoiesis of indeterminate potential. CHIP is one of the ways with which clonal hematopoiesis can manifest. This is a sort of a representation of the evolution of CHIP clones. As you can see, it could be any hematopoietic stem cell that in its lifetime gives rise to a lot of progeny. Some of them are going to acquire some random mutations that just persist along and pass out. There are instances where one of those mutations could actually be resulting in a set of clones that have some features which make them sustain throughout the generations, then go on to be a part of tumor cells. Now, mind you, these are not driver mutations for tumor. These are just mutations, maybe increasingly thought of as a pre-driver mutation. With that said, why are we interested in studying CHIP? Well, clonal hematopoiesis in itself can impart fitness advantage to these clones. Now, this can happen via increased self-renewal or increased rate of proliferation or even reduced cell death. And the incidence of CHIP and prevalence of CHIP increases with increase in age. For example, in individuals aged over 65, the rate of detection of these clones is between 10 to 15%, and it increases by greater than 30% when we are looking at individuals over 80. The key reason why we are interested in CHIP is that these mutations somehow happen in leukemia-associated genes or leukemia driver genes for most of the times, particularly DNMT3A, TET2, and ASXL1. Also, some of the genes most mutated in CHIP are JAK2, P53, BRAF. So the question here is, we know these, by definition, CHIP does not have any hematological malignancies, but why are these mutations happening in these leukemia-associated genes? So the fate of these mutations are actually dictated by two phenomena. There is an interplay between the fitness advantage that these clones gain that I explained before, and also there is a preferential selection factor for specific mutations. What I mean by that is in individuals who are older, primarily these chip mutations are seen in genes associated with spliceosomes. For individuals who have had any history of smoking, most commonly the chip mutations occur in ASXL1. And individuals who underwent cancer therapy, then pretty much the genes of double-stranded DNA repair are the ones that show these chip mutations. Now, numerous studies have detected chip prior to chemotherapy in patients. So this suggests that it might play a role in therapy-related myeloid neoplasms. Also, the overall survival rates reduce when there is an association of chip. And chip is also strongly correlated and associated with cardiovascular disorders. So this means understanding the inflammatory effects of chips, particularly on a tumor microenvironment, can help us figure out if it is promoting chemoresistance or metastasis. With that said, there was a prior research that was conducted in our lab looking at over 100,000 solid tumors, and they found an interesting phenomenon where the rate of detection for TET2 was higher than that of DNMT3A. Well, why is this interesting and relevant? The plot on the right actually shows a recent study, but this is the overall trend that has been repeatedly shown in all the publications that have studied chip, where the most detected, most commonly mutated gene is DNMT3A, followed by TET2. But we see a reversal inside the tumors. So this was fascinating, and this prompted a couple of research questions. The first one was, can we reliably detect chip inside solid tumors? Because most of the studies look at blood. And if we actually can, then the chip that we see in the normal blood, is it going to be different from solid tumors, given what we saw in those 100,000 tumors? So as I mentioned before, chip is often detected in peripheral blood samples using typical somatic variant callers, MUTEC2. And in order to detect chip in tumors, we decided to use sequence sample pairs, namely primary tumor sequence and matched normal blood sequences. The most abundant source of these sequence pairs are actually high throughput, low depth whole exome sequencing datasets, namely TCGA and Orion. So with all of this information and the reasoning behind why we want to pursue chip, we came up with two specific aims for my dissertation. The first one was to develop a robust computational pipeline that is capable of detecting these low abundant mutations in tumors and blood sequences, and then on to build a scalable framework from this pipeline, which is actually capable of handling tens of thousands of samples and millions of variants in a reproducible manner. Before I explain the approach that we took, I would like to talk about the relationship between read depth and rates of detection. Here, I'm showing the total depth distribution for 32 samples, whole exome samples from TCGA's lung investigation. And as you can see, the average depth for these exome sequencing samples is around 100 reads. Now, why is this important? Well, here I'm trying to detect a variant which has an allele frequency of 20%. As you can see from the plot on the left, the probability for that variant call to be a true variant, right? We are essentially only trying to differentiate what is a true variant and what is a sequencing error at this point. So for it to be a true variant, the probability of that increases with increase in total depth. And the confidence intervals also, it starts out pretty wide, but then it narrows as we have an increase in total depth. Now, let's sort of switch gears and look at the same plots, but this time for an allele frequency more in the realm of chip. The lowest typically detected allele frequency for chip is 2%. Here you can see that for the same depth of 50, the probability that that variant could be true drastically reduces. And as the probability, as the total depth increases, the probability will also increase. So what this tells us is, without an understanding of the total reads that we are looking at, making calls on allele frequencies alone to determine what variant could be chip is not informative, and we might be looking at false positives that are arising purely out of background noise. So there are two core tools that are used that enable this study. The first is MERIT, and as Rowan mentioned, it is a comprehensive pipeline designed by Mohammed in our lab, and it's designed for in-depth quantification of erroneous high-throughput sequencing calls. And by detecting all the variants in the sequencing data, MERIT actually acts as an agnostic variant caller where it will not filter anything out. It's up to us to determine what those filters are going to be. Then followed by Backtrack, a computational method also developed in-house in lab to discern low abundance mutations from background error in sequencing data. Backtrack was originally designed for high-depth sequencing, but it actually works for our purposes too. Now, let me draw your attention to this one table. Here, we are looking at a single mutation, which is a hotspot mutation in DNMT3A, known as R882H. It happens in chromosome 2, where DNMT3A gene is, and it's a C to T conversion. What we are looking at are the alternate depth and total depth for this mutation across 32 samples. These are 16 pairs of primary tumor and matched normal samples from whole exome sequencing data in TCGA lung study. Now, what I want to emphasize here is that each read of an alternate allele that we see in any given sample here can be treated as a coin toss of a head or a tail. There are odds of us seeing it or not, which essentially boils down to be a binomial probability for observing this alternate allele C to T in this position in that sample. And this can be computed for every sample. Now, Backtrack helps us quantify these odds such that what are the odds of, let's take, for example, the sample highlighted in green here, where we see one alt allele out of 55 in the context of all the other samples having the same alt depth distribution. What I mean by that is, let's sort of switch to the sample highlighted in blue. This paper by Shi et al., where they spoke about these hotspot mutations in CHIP, actually detected this R882H in the pair shown here in blue and yellow. Blue would be the normal blood sample, and yellow is the primary tumor pair of that sample. And you can see that in blue, there are 15 alt reads out of 47 total reads. And the probability for observing this happens to be somewhere, according to Backtrack, in 10 to the power of minus 24. And the false discovery rate for this is, again, really high in the sense that it's probably a really true mutation. The odds of it being false is very low. Now, when we try and look at the same probability values in FDR for all the other samples that are shown in this table, we can see there is only one other sample, the one in green, that actually shows up this read. So we might be tempted to say, 1 out of 55. We saw that, total depth of 50 is actually good to be able to determine if it's a true read or not. And it is showing a read where none of them are. So most likely it could be a true mutation. But if we look at the false discovery rate computed by Backtrack, it's not much different from what we see for all the other samples that do not have any reads. This tells us that, what we are looking at is most likely a sequencing error and therefore background noise. And the true signals are only from the pair in blue and yellow. So this is the whole crux of using MERIT and Backtrack. We cannot use any other variant caller for our approach because most variant callers might filter out that green read, that 1 out of 55, due to either low quality or low call, or and also we won't be able to capture all the information of, no reads. But because MERIT is designed in the way to be agnostic and it calls any and all variant that it sees, we can use that information and find out the distribution of the alt reads across all the samples. Then we can use Backtrack and then create noise models for each and every mutation, which will help us identify what are putative CHIP mutations. So now let me talk about some of the challenges in this approach. First is performing variant calling itself on tens of thousands of samples to generate a list of all variants using MERIT. We decided to use a pan-cancer approach in TCGA, therefore that's the first challenge. And the second is, how do we compile that information that comes out of MERIT and generate these noise models, again, on these millions of variants using Backtrack? So the data set that we chose for this study is the Cancer Genome Atlas, and it's a pan-cancer study, and we have 7,797 individuals, and we are looking at their molecular data, particularly the exome sequencing data for primary tumors and matched normal pair. The reason why we have 7,797 individuals is because of this filter. So we did not want to look at any individual sample that was FFPE preserved. And as I mentioned, we are looking at pairs, and we are looking at TCGA. And we included a caveat here such that we wanted to look at investigations that had at least 100 samples. Now, why did we do this? If I try and go back to this table here, you can see that a lot of these samples are having 0 reads, and some of them are, there's just one that has 1 read, and then the 2, which we know to be true mutation, having higher reads. Now, this background model is only as robust as the amount of information available to it. So how can we add more information to this particular position? Either we can do high-depth sequencing, which we know we don't have. We are working in the realm of exome sequencing, and we know the total depths are going to be around 100. So is there any other way to add more information to this particular position? Well, then we thought we are not interested in adding more information to a particular sample, but we are interested in adding more contextual information to the mutation. So we can do that by actually increasing the number of samples within this table. For example, from 32 samples, we can make this table to be 100 samples long. So what that would do is it will add more information to Backtrack and generate a much better noise model, and it will in turn affect the way the FDR values are calculated, and it will help us in determining more meaningful results out of our calculations, which is why we had this caveat of, we have this cutoff of at least 100 samples we want. And these are exome sequencing BAM files. Now, the first big data challenge here is a typical TCGA exome sequence file is somewhere between 15 to 25 gigs in size. And for 7,797 cases, which plays out to be around 18,000 files roughly, the total size is around 360 terabyte. Now, to sort of visualize how big 360 terabyte is, we can conveniently store all the books that are in the Library of Congress, which is more than 25 millions, by the way, in an uncompressed manner, and still have space left over in that much space. So clearly, this is not something that we can keep at one place or even conveniently download and upload and access. So with that in mind, the infrastructure for this project was determined to be first, we will use the Rutgers high performance cluster, AMRL, in order to design the pipeline. And we would deploy this pipeline on CGC, which is an excellent cloud computing resource funded by NCI, particularly for the aspect of facilitating access and research on large cancer datasets like TCGA. Some of the other tools that were used for the study are Samtools, BCF tools, SnipFN, SnipSif to annotate the VCF files or the variant call files, Docker to create the container for Merit, which can then be deployed on CGC, and Python 3 for general scripting needs. This project was designed in 3 phases. The first phase was to test the validity of this approach. We wanted to make sure if we can actually detect CHIP through this method. And that's where the table that I showed you comes from, where we looked at 32 samples and we looked at one particular mutation, and we saw if we can call it. The second is to build and benchmark this pipeline using roughly 200 TCGA lung cancer sequences, also on Rutgers HPC. And finally, to deploy the pipeline for CHIP detection on the pan-cancer TCGA dataset on CGC. So when I say the pipeline, this is how it looks at. We start with the exome sequence file. Then we sort and index those BAM files. Originally, we decided to split these BAMs by chromosomes in order to achieve 2 things. 1, it reduces the computational load per task. And 2, it gives us the ability to massively parallelize the task. So from running one sample for 30 hours, we run 24 instances of the same sample. Each of them might take 2 hours, but 24 parallel instances would mean we are achieving what we would take in 30 hours in 2 hours. Then we perform variant calling by Merit. And Merit actually is a 6-step process. It generates the pileup file, it detects variants, creates variant call files, it can annotate variants, And in our case, we chose to do it with dbSNP, Cosmic, and also it extracts base quality scores from the pileup and adds to the variance. Then we have our own filter criteria. Namely, first one would be the depth, as have I hopefully I have stressed by now the importance of having sufficient total depth. Then we check for strand bias calls, we check for quality, and in this step, we actually initially decided to remove calls from dbSNP, thinking they are most likely going to be germline, and since we are interested in somatic mutations, that could reduce the number of mutations that we are looking at. Following this, backtrack would be ran on all the variants that pass this. So in phase 2, as I was building the pipeline and optimizing it and computing resource requirements, what we found is that step 4 of merit, where actually after the variants are called, merit goes back to the pileup and it computes the quality scores and adds it to the variance. That was the bottleneck of this entire toolkit. It's such that on average, it took around 13.5 GB of memory and ran for roughly 20 hours. Now, we have roughly 20,000 samples at the time. It was 18,000 is the final number which we ended up with. But even if we run 200 parallel tasks, where each task is going to have 16 GB of memory and roughly 200 CPU cores and run for 20 hours, it would still take us 2,000 days to process all of these samples just for step 4. And if you were to go with the maximum runtime, it could take up to 3,000 days. As you all would agree, this is not realistic in the timeline of a grad student or any project for that matter. So we wanted to go back to board and find out how we can improve this. So we did 2 things. One, Merit was originally written on Python 2.7. So we decided to upgrade it to Python 3. There were optimizations that were carried out on Python 2 to 3 migration itself, which meant that some of the runtime requirements were made lower. The memory requirements became smaller. However, we also looked at the data structure of Merit in itself and made changes such that now, for example, chromosome 1 here, which is the biggest chromosome of the 24, which did not run even after 3 days, taking up roughly 50 GB of memory with previous version of Merit, actually now ran in under 10 minutes and with half the memory. And as you can see, for even smaller chromosomes like chromosome Y, the gains are even bigger. So this meant that now we can actually implement this pipeline in a realistic timeline. So for the same 20,000 samples, assuming 200 parallel tasks from 5 years, now for entire Merit, right, not just for step 4, it would only take 4 months. So with this, excuse me a second. Now I want to talk about the quality filters that I mentioned before, that assuming we run Merit and we get all the calls, how are we going to filter them? And how does the filtration look like before we run Backtrack? So here, we, as part of phase 2, we called Merit on all the variants in TCGA lung cancer dataset, which would be LUSC and LUAD. It totaled 1,535 samples. And Merit identified close to 3 billion unique variants across these 1,535 samples. We all can agree that's a ridiculous number. So when we applied each step of the filter, particularly we removed strand bias, a lot of these went away, which we expect, right? Not every single call is going to be true, which is why all variant callers have their own inherent filtering mechanism to try to weed out most of the garbage calls. So after strand bias was removed, we had 200 billion variants, which was a huge reduction. And then we applied our depth cutoff, and that meant we had 125 million variants, quality scores, and finally, after removing everything in dbSNP, we ended up with roughly 65 million variants. Now, this is a significant reduction from all the initial variants that we had, but this is still really high. But I want to talk about how the Backtrack pipeline was set up on AMREL. So in order to generate the tables that you saw before, those tables had to be generated for every single mutation. So effectively, for 65 million individual tables had to be created. And this is how we went about it. we created two tables, two set of tables actually. One would be for total depth, one would be for the variant depth for each mutation. Excuse me. So each of these tables will be created for each chromosome. That's why you see 24 on both sides. Now the total depth table and the variant depth table is going to have for every sample what is the total depth and what is the alt reads for every single variant in that passed the previous step. So here the length of these tables would be such that there will be 65 million rows across each of the 48 tables. And after this, we combine each of these rows into one table and create 65 million individual files. Now these files would be put in a bunch of folders, such that here we decided to have 2000 files per folder. The reason is, I found out anything over 50,000 files in a folder, and Linux bash cannot even run ls, ll, none of those things, right? It's too many files for the system to handle. So we have to limit those files. So we sort of hit 2000. So there would be a bunch of such files. Each files will have 2000 such variants, and then on each of those simultaneously we'll try and set up backtrack. Here's the second big data challenge. As I briefly mentioned, the problem with the Linux file system unable to handle a large number of files. Another thing that I discovered through this is high performance clusters actually have not just limitations on the volume of files you can store, but also on the number of files that you can have. What I mean by that is, for one single run that I just described to you for the TCGA lung cancer data of 1,500 samples, we create 1 million files. And at one point, I had 50 million files in my system. They were totaling only like 10 terabytes. So storage-wise, I was okay. But I could not move, read, write any of the files in the system because I had way too many files. So then, a combination of the necessity to reduce the number of files and also recognizing that 65 million variants from one tumor investigation is not reasonable to parse through. So what we did is we tried to come up with a reasonable way to address this. And what we have found is most of the publications that investigate CHIP, even though they have access to whole genome, whole exome data, they usually focus only on genes that are known to have associations with CHIP. So from 4 different papers, we created a superset of 113 CHIP-associated genes and decided to focus only on those. Now, what that looked like is we had these intersection of 3 sort of positions coming into play. On one hand, we had in green all the unique variants that are called by merit after all the filtration. So if you're looking at lung cancer, that would be 65 million variants. The other hand, we have these 103 gene positions. So we look for overlaps. And furthermore, we also look for positions that are originally covered by the SureSelect probes that were used for this exome sequencing in itself, because we all know off-target effects exist, and we did not want to have those variants inflate the numbers that we had. So ideally, we looked for intersection of those variants that are shared by these 3 groups. And those were the variants that we used for our analysis. So now let me talk about CGC and Phase 3. So here, as I mentioned, we looked at 16,500 samples, roughly from 7,797 cases. These span across 21 tumor types and 27 investigations. And now Merit 2.1, which is on Python 3, was deployed on CGC. And we only targeted the regions in the 113 chip-associated genes. Here we retained the dbSNP variants. The reason was earlier when we were testing it on Phase 2 without dbSNP, we found that the hotspot mutation that we were looking for, the R882H, was filtered out. So that's when we realized some of the somatic mutations that are also hotspot mutations have a dbSNP RSID. So we decided to keep them and call all of them. After this, we removed any multi-allelic variants before we ran Backtrack. And in the interest of time for my dissertation project, we chose to focus on 4 investigations: LUSC, LUAD, BRCA, and BLCA. This totaled roughly 4,400 exome sequencing samples. After this, once we run Backtrack, what we did is try to again whittle down the total number of variants by trying to look at a putative chip variant set. We did that by first try to remove the sequencing artifacts. So anything that had, we only looked at the 10th percentile of the false discovery rate cutoff to eliminate most of the noise. We removed indels, and any mutation that had a total read depth of greater than 500 was also eliminated because they are mostly due to PCR amplification errors. And in order to filter out germline calls, we looked at a cohort frequency of 3%. So what that means is if a variant is seen in more than 3% of the samples in that investigation, then we think it's most likely a germline call and we remove it. And also we used allele frequency cutoff such that anything greater than 40% is most likely to be a germline call as well, and it was removed. So after all this, I want to sort of share some data from what we found. The first is trying to see if we are actually detecting variants across all the samples. And as you can see here, these are the 4 investigations. The one in blue are all the unique variants that are called by merit, and they are called in every sample. Post Backtrack, we still have variants in every sample, but when we apply the Backtrack filters from the previous page, then we actually start reducing the number of variants and in turn the number of samples that actually show these mutations. Now, when we call these tumor mutations, they typically tend to follow along these axes in 3 groups. There are going to be mutations that are called in both tumor samples and blood. There are going to be mutations that are seen only in blood, and there are mutations seen only in tumor. So the table on the right shows that across these 4 distributions, what were the number of mutations called across each of these groups. And what's interesting is, as you can see, blood-only mutations were really rare in our cutoff, and of that, they were actually shared between these groups. So that was interesting. So now let me bring it back to one mutation in particular, which is the R882H again. And here we are trying to see if we can detect that DNMT3A hotspot mutation in bladder cancer. And we actually see it in a sample, but also in the blood tumor pair too. So in blood, it's a 36% allele frequency, really good, robust. And in tumor, less than 2%. Now, this is where that mutation is. And what is interesting about this is that bladder cancer was not one of the tumor types originally investigated in the study that actually identified this hotspot mutation. They identified this hotspot mutation in lung cancer, and they also had glioblastomas, and they detected there too, but they weren't looking at bladder cancer. But we can see that that hotspot mutation is also seen in a bladder cancer individual. Now, the same approach was applied to investigate breast cancer chip mutations on a collaborator study from Moffitt, where we tried to look at blood in a retrospective way. So what I mean by that is we had access to blood from patients which was collected before treatment and after treatment. So we said, okay, can we detect chip in this cohort? And the sequence depth here is actually really good. I was jealous. It was 5,000x. And when we looked at 10 patients as a pilot study to try to determine the direction this can take, we were surprised to see clonal hematopoiesis mutations detected in 7 out of 10 patients, which was fantastic news. And what's interesting is that these mutations did not always increase in allele frequency post-therapy. Most of them did, but particularly TET2, actually after therapy, the allele frequency was reduced. So this told us that, the pipeline is robust. It can help us investigate these longitudinal / retrospective questions. And so we are furthermore expanding the study to hundreds of samples, same deep sequencing, and trying to apply this protocol and come up with an early diagnostic measure. So, okay, I've spoken to you for a good 30 minutes, I think, at this point, about CHIP and my project and merit. So what's the takeaway message from all this? What's the utility in all of this? Well, early this month, NIH actually allocated a huge sum of money in particularly investigating somatic mosaicism. Now, somatic mosaicism or somatic variance is an area where this pipeline thrives. Those are low abundant mutations. Those are going to be small in number. This pipeline is built to track those mutations. And we are working on the public release of the beta app on CGC for everybody. So Merit is already on CGC, and I used Merit to call variants for my work, but I used it as a private app. So now we've sort of cleaned it up a little bit, added some description, and trying to get more users to test it before we do the full release. So we can use Merit to answer some of those somatic mosaicism questions. We can also go ahead and quantify the level of lymphocytic infiltration for the detected CHIP mutations from my study by looking at the RNA sequencing data in TCGA. We can look at the tumor-only signals that we have, try to see are there stromal mutations. We can also apply this exact framework, as I showed in Moffitt data, to Orion network. As I mentioned before, we can also conduct longitudinal / retrospective studies that can shed light on the clonal progression and evolution of the somatic mutations. We know these mutations, CHIP and clonal hematopoiesis, is interesting, but we don't know what exactly they do. And here is an opportunity and yet another tool to try and make sense of it and understand the biology better. With that, I would like to thank my PI, my lab members at Rutgers. Now I'm a previous member, so that's fun. I also would like to thank my dissertation committee, who are very supportive, Rutgers HPC professional, and particularly Rowan, Sarah, and Dave at CGC, who sort of, I did not explain the level of support I would get from them, in the sense that I expected it to be purely technical, but they actually were significantly helpful in me getting through the last moments of my PhD, where there was mental health support. Like, they just listened to me rant about something that isn't working. That helped a lot. And the life of a computational biologist is mostly really lonely. So to have people who have gone through that pain and to share that with them was, I appreciate it a lot. I thank you all for your support. And with that, I'll be happy to take any questions.